There's a fungus that makes flies die in a ritual-like pose. Another hijacks cicadas with mind-altering chemicals similar to amphetamine. And some force ants into a final act of perfect precision. These are the fungi that bend nature to their will, starting with a fungus that positions flies for something far more sinister than a normal death. Entomophthalma muscae. This parasite specializes in flies and has evolved a reliable way to spread its spores by forcing its host into a very specific final act. The infection begins when a fly comes into contact with a fungal spore. The spore sticks to the fly's body and soon penetrates the protective exoskeleton with a combination of mechanical pressure and enzymes. Once inside, the fungus spreads through the insect's body, consuming nutrients and gradually replacing tissues. For a few days, the fly behaves normally, flying and feeding as if nothing has happened. This period allows the fungus to multiply without drawing attention. Then the parasite begins to alter the fly's behavior. Instead of continuing its usual activities, the infected fly seeks out an elevated, exposed position. Often this means the upper part of a window, a plant stem, or a wall. There it fixes itself in place. The legs grip tightly, the wings spread, and the abdomen lifts upwards. Scientists call this posture the death pose. It is not random. The position gives the fungus maximum opportunity to launch spores into the air once the host dies. After the fly has been locked in this position, the fungus completes its life cycle. That's horrifying. The system is efficient. In environments with many flies, outbreaks of Entomophthalma can kill large numbers in a short time. The story of Entomophthalma muscae is a reminder that behavior in nature is not always controlled by the individual. In this case, a fungus has found a way to determine not just where its host dies, but how it dies, turning a single fly into a distribution system for countless spores. Next, a parasite that directs ants with an accuracy that seems almost unreal. Ophiocordyceps unilateralis is the most iconic. It infects carpenter ants in tropical forests and forces them into a sequence of actions that ends with their death in a very specific location. For the fungus, this precision is the difference between survival and extinction. The process starts when a foraging ant encounters fungal spores on the forest floor. These spores attach to its body and penetrate the tough exoskeleton using enzymes and mechanical pressure. Once inside, the fungus spreads cell by cell, drawing nutrients from the host and growing throughout the body. During this early stage, the ant appears perfectly healthy. It continues working in the colony, unnoticed by its nestmates. This symptom-free period allows the infection to progress without being detected and eliminated. After several days, the fungus exerts control. Instead of returning to its nest, the infected ant is compelled to leave the safety of the colony. It descends from the canopy to the understory, where it climbs onto a small plant. At a height of about 25 centimeters above the ground, the ant performs the last action of its life. It bites down on the central vein of a leaf with such force that its jaws lock permanently. Scientists call this the death grip. The ant dies fixed in place, while the fungus begins the next stage of its life cycle. That's scary. From the ant's body, fungal tissue breaks through, eventually forming a stalk that emerges most often from the back of the head. At the tip, a capsule develops, filled with spores. When mature, it releases them into the environment below, exactly where other ants from the same colony are likely to pass. The parasite is not only controlling the ant's movements, but also ensuring the final location is perfect for its own reproduction. This ability is ancient. Fossil evidence from Germany shows the marks of an ant death grip on a leaf dating back around 48 million years. In the middle, a parasite that doses cicadas with chemicals similar to amphetamine, Massospora cicadina. This parasite turns the insects into unwilling carriers, keeping them active while their bodies are being destroyed from the inside. Infection begins when cicadas come into contact with fungal spores lying dormant in the soil or released from previously infected individuals. Once inside the body, the fungus grows rapidly. Within a few days, it replaces large parts of the insect's abdomen. The infected segment eventually breaks away, leaving behind a white, chalk-like mass of fungal spores where the rear of the cicada should be. Despite losing critical body parts, the cicada remains alive and continues moving, flying, and searching for mates. The most disturbing part of the infection is the change in behavior. Massospora produces psychoactive compounds, including chemicals similar to amphetamines. These substances alter the cicada's nervous system, driving it to hyperactivity and compulsive mating attempts. Males may attempt to mate not only with females but with other males, dramatically increasing the number of contacts. Each interaction spreads spores which stick to new hosts and continue the cycle. The white mass of fungal tissue is visible to the human eye, often replacing nearly a third of the insect's body. Yet the cicada continues to act as though it is intact, attempting flight and courtship. It's crazy, but the fungus has also attracted attention because of the chemicals it produces. The presence of amphetamine-like compounds in a natural parasite is unusual, and researchers are studying how and why the fungus makes them. The prevailing theory is that the compounds directly manipulate host behavior, keeping cicadas active and more likely to interact with others, which ensures maximum distribution of spores. For humans, the fungus poses no danger. The compounds it produces are not present in sufficient amounts to affect people who handle infected insects, and the parasite cannot develop in mammalian hosts. Its significance lies in the way it demonstrates the strategies parasites can evolve. Instead of simply killing its host, Massospora cicadina has found a way to keep it alive, chemically manipulate its behavior, and use its body as a vehicle for reproduction. Then comes a parasite that keeps flies alive while secretly replacing their tissue with its own. Among the strangest parasitic fungi ever studied are species in the genus Strongwellsii. They infect flies, but instead of killing them quickly, they hollow them out from the inside while leaving them alive and active. The result is a host that looks intact at first glance, but is already serving as a vessel for fungal reproduction. The infection begins in the usual way. A spore lands on a fly and penetrates the exoskeleton. Inside, the fungus grows rapidly, filling the abdominal cavity. Unlike other parasitic fungi, however, Strongwellsii does not force the host into a single death pose. Instead, it opens a hole in the fly's abdomen, creating what scientists call a spore window. From this cavity, fungal spores are continuously released into the air. The most unsettling aspect is that the fly does not die immediately. For days, sometimes more than a week, the infected insect continues to fly, feed, and interact with others as though nothing were wrong. All the while, spores are streaming out of its body, settling on surfaces or directly onto other flies nearby. In this way, the parasite transforms its host into a mobile distribution platform, able to spread spores far more effectively than a stationary corpse. Researchers have described this phenomenon as active host transmission. The evolutionary logic is clear. It's scary. By keeping the fly alive, the fungus gains more opportunities to reach uninfected individuals. A stationary cadaver can only infect those that pass nearby. A living, mobile insect, on the other hand, carries the infection into new areas, including clusters of healthy flies. For a parasite, this strategy is efficient, even if it appears grotesque. A fly with a hole in its body, still flying through the air, is one of the most vivid examples of how far fungi can push these boundaries. And finally, a fungus that turns European forests into graveyards of infected ants, Pandora formicae. Unlike its tropical relatives, this parasite targets wood ants and creates a phenomenon that looks like a miniature battlefield. Clusters of ant corpses attached to grass and twigs, each one a victim of precise manipulation. The cycle begins when an ant comes into contact with fungal spores in its environment. As with other entomopathogenic fungi, the spores penetrate the cuticle and spread inside the body. The ant continues its normal activity for several days, showing no obvious signs of infection. During this time, the fungus establishes itself within the host, feeding and spreading throughout the tissues. When the parasite reaches the right stage, it compels the ant to leave its colony and climb vegetation. Unlike Ophiocordyceps, which prefers leaves in tropical forests, Pandora formica typically forces ants to attach themselves to blades of grass or low plants. The ant bites down and remains fixed in place, entering its final moments. Soon after, the fungus consumes the rest of the body and grows outward, producing spores that will drift down onto other ants in the colony below. Scientists studying Pandora formica have also noted its precision. Infected ants are guided to positions where humidity and temperature favor fungal growth. Even the orientation on grass stems appears consistent, maximizing the chance that spores reach new hosts. It is a system refined by millions of years of evolution. In the broader context, Pandora formica demonstrates that the phenomenon of zombie ants is not limited to tropical jungles. It is present in Europe as well, adapted to local climates and ecosystems. The same fundamental principle applies. The parasite does not simply kill, it directs when and where death occurs to optimize its own survival.